Hello friends, welcome to my channel Pharma Dive. This channel is only for educational purpose and aims to provide valuable information. So if you are ready to dive into the world of pharma, hit the subscribe button and join the journey. Let's explore, learn and gain together. Today I am going to talk about stability studies in pharmaceutical industry. Have you ever wondered how medicines stay safe and effective till their expiry date? That's where stability studies come in. Stability is the capacity of a drug product to comply with established specifications throughout its assigned shelf life when stored under labeled conditions. Stability studies evaluate how the quality of a drug substance or a product changes over time. These studies are conducted under the influence of environmental factors such as temperature, humidity, and light. They are essential for determining the shelf life and appropriate storage conditions of the product. Stability studies are carried out in accordance with ICH guidelines. The objectives of stability studies are to establish the product's shelf life, also known as its expiry date. These studies help recommend appropriate storage conditions to ensure the product remains stable. They also assist in determining suitable packing components that protect the product from environmental factors. Stability studies ensure that the product maintains its quality, safety, and efficacy throughout its life cycle. Additionally, they play a crucial role in supporting global regulatory submissions. Let's see what is stability protocol. A pharmaceutical stability protocol is a formal approved document outlining the scientific ICH compliant plan for testing drug substances or products over time. It defines the study type, including long-term, accelerated, intermediate, or photostability testing, product details, and batch selection information, storage conditions and duration, the exact container closure system, sampling time points, test parameters, and acceptance criteria, ensuring quality and determining shelf life. How do scientists predict when a medicine will expire? The answer lies in smart data analysis. Shelf life determination in pharmaceutical stability studies establishes the time a drug remains safe and effective under defined storage conditions, typically ending when the degradation products exceed limits or potency drops below 90 to 95%. Shelf life determination is based on analysis of stability data collected over time. It involves the statistical evaluation of degradation trends to understand how the product changes under the different conditions. The shelf life is assigned only when the product consistently meets the established specifications throughout the studied period. Let's understand stability testing parameters. Critical quality attributes in stability studies are physical, chemical, biological, or microbiological properties that must remain within defined limits to ensure drug safety, potency, and quality. Commonly monitored critical quality attributes in stability studies are appearance, assay, degradation products or impurities, dissolution, pH and viscosity, moisture content, microbial limits, container closure integrity, and particle size or particulate matter. Testing frequency. Stability testing isn't done just once. It's a carefully planned timeline to track a medicine's quality over years. In long-term stability studies for a product with at least 12 months of shelf life, samples are withdrawn and tested at predefined intervals. During the first year, testing is conducted every 3 months, that is at 0, 3, 6, 9 and 12 months. In the second year, samples are tested every 6 months at 18 and 24 months. Thereafter, testing is performed annually at 36 and 48 months and so on. There are different types of stability studies. First one is long-term studies, which are real-time storage conditions conducted over 12 plus months under recommended storage conditions to set the expiration date. Second one is accelerated studies, which are high-stress conditions to predict stability. They are conducted at higher temperatures and humidity for 6 months to predict rapid degradation. Next one is intermediate studies. conducted for minimum of 6 months to provide more data if the accelerated study shows significant change. The suggested storage condition for storage of products at room temperature are given in the following table. The suggested storage conditions for storage of products in refrigerator are as follows. The long-term studies carried out at 5 degrees Celsius plus or minus 3 degrees Celsius for 12 months, and the accelerated studies are carried out at 25 degrees Celsius plus or minus 2 degrees Celsius at 60% relative humidity plus or minus 5% relative humidity for 6 months. The suggested storage condition for storage of products in freezer are as follows. Long-term studies are carried out at minus 20 degrees Celsius plus or minus 5 degrees Celsius for 12 months, and accelerated studies are carried out at 5 degrees Celsius plus or minus 3 degrees Celsius for 6 months. Other types include photostability, that is light exposure studies, stress testing or force degradation, in-use stability studies, freeze-thaw stability studies, and hold time studies. Did you know medicines are tested differently depending on the climate they will be used in? That's where ICH climatic zone come into play. The International Council for Harmonization classifies the world into different climatic zones for stability studies based on temperature and humidity conditions. Zone 1 represents temperate regions. Zone 2 includes subtropical and Mediterranean regions. Zone 3 represents hot and dry climates. Zone 4A represents hot and humid regions, and Zone 4B represents hot and very humid climates. These climatic zones helps ensure that stability studies accurately reflect real-world storage conditions across different parts of the world. In case of excursion occur in set temperature and or relative humidity of stability chamber due to any reason like breakdown, power failure, door kept open for longer time, etc., it can be categorized as per provided table and action to be taken as specified. Stability chambers are specialized ICH compliant environmental units used in pharmaceutical studies to simulate various climatic conditions, ensuring drug safety, efficacy, and shelf life determination. They maintain specific temperature and humidity conditions. They are equipped with continuous monitoring and alarm systems to ensure that any deviation is immediately detected and controlled. What is significant change? A significant change in pharmaceutical stability testing is defined as substantial, unacceptable alteration in product's physical, chemical, or microbial quality, violating ICH Q1A R2 guidelines. If long-term studies are conducted at 25 degrees Celsius plus minus 2 degrees Celsius at 60% RH plus minus 5% RH, and a significant change occurs at any time during 6 months testing at accelerated storage condition, then additional testing at intermediate storage condition should be conducted and evaluated against significant change criteria. It indicates that the product no longer meets specification, often marked by greater than or equal to 5% assay change, triggering a need to investigate, reduce shelf life, or modify storage conditions. Some examples of significant changes are: first one is assay, a 5% change from initial potency value or failure to meet acceptance criteria. Next one, degradation products, any impurity exceeding its acceptance criteria or threshold. Next is physical appearance. Physical failures such as discoloration, cracking, capping in tablets, or phase separation in emulsions. Next one is functionality. Failure to meet dissolution specifications for 12 dosage units. pH falling outside the appropriate range. And microbiology. Failure of sterility or preservative effectiveness test. ICH guidelines are: ICH Q1A R2 is the core guideline that outlines stability testing requirements for new drug substances and products. ICH Q1B focuses on photostability testing to ensure that products remain stable under light exposure. ICH Q1C provides guidance on stability testing for new dosage forms, while ICH Q1D explains bracketing and matrixing approaches used to reduce the number of samples without compromising data quality. ICH-Q1E deals with evaluation of stability data to establish the product's shelf life. ICH-Q1F was originally designed for stability data requirements in climatic zones 3 and 4, but it has now been largely withdrawn and incorporated into ICH-Q1A, making the framework more streamlined and globally harmonized. Let's see regulatory requirements. Stability studies in pharmaceuticals are mandatory harmonized regulatory requirements that primarily follow the International Council for Harmonization Q1A to Q1F guidelines. These studies are designed to establish retest periods, shelf life, and recommended storage conditions based on data generated from testing at least 3 batches. Global regulatory authorities such as US FDA, European Medicine Agency, and World Health Organization rely on stability data to evaluate product quality. Stability studies are mandatory for product approval and play a crucial role throughout the entire product life cycle, ensuring ongoing safety, quality, and efficacy. Conclusion: stability studies are critical for product life cycle management. They help to determine the shelf life and storage conditions, ensures compliance with global regulatory standards, and it is a key tool for maintaining pharmaceutical quality. Thank you everyone for your time and attention. If you like this presentation on stability studies, then please like, share and subscribe my channel Pharma Dive.